Hello, my name is Tomasz Kurowski. I'm a lecturer in bioinformatics at Cranfield University. And I know most people at this conference are mostly working with, for example, human data. I've seen a lot of microbial. I didn't see all that many people working with plants, although that is mostly what I do. So I work with tomatoes, raspberries. Recently, I've been working with tea, or rather is really the plant molecular biologists that I work with that deal with those. I deal with the data. So just to give you a brief overview of what the biological problem is that we're dealing with here. As an example, tomato is the one genome that we've been dealing with the most. It's well characterized. There is a high quality reference genome for it. 12 chromosomes, well behaved, diploid, around 1 gigabase in size, and there's hundreds of sequenced, resequenced cultivars and closely related wild species available for it. That's our phenotyping facility, and that's raspberries in the lower right. And tomato has quite a few wild relatives in South America. There's all across the west coast and Galapagos Islands, and many of these can be relatively easily crossed into the cultivated tomato and can be an important reservoir for useful traits. And this can be done in a very organized fashion. We have these introgression lines being developed for tomato when you introduce bits and pieces of the wild genomes into elite cultivars. But this has also been happening naturally. This has also been done by breeders previous decades, before we've had this sort of ways to sequence and investigate sequences as we do now. So very often what happens is those introgressions exist there, though that foreign DNA exists there, and there are interesting traits associated with it, but we don't necessarily know exactly where they came from, where they are placed across the chromosomes, and we will get a project with a industrial partner that will say, we have this very nice phenotype in this line here, we have no clue where it came from. So we will be investigating that. And one example of it is a good few years ago now, we've had a PhD student named Dimitrios working on basically figuring out what causes this distinction between these two phenotypes in inflorescence branching in tomatoes, in a particular line of tomatoes, really. And he, across his three-year project, figured out that, okay, there is this 2 megabase genome, region of the genome where the causative gene for this is located. And we've managed to figure out afterwards, comparing to all those already published genomes, that actually this small section of the genome comes from the Solanum galapagense wild species. And who and when introduced it there, maybe it was natural, we couldn't really tell. But it was something that we could identify. So someone's done this, maybe nature did it. And the comment that the supervisor made then to Dimitrios as well, we're playing the same game, we're just seeing the cards now. And the way this was visualized was using a tool called Tersect Browser, which the plant molecular biologists were very impressed with, which basically generated this sort of heat map of genetic distance to your particular genome. So anything close is lighter in color, anything more distant is darker in color. And when you zoomed in just to that interval, you see that this cultivar clusters with Solanum galapagense. And to the left of this, you should be also seeing a phenotype, a phylogenetic tree, sorry, but this tool generated it separately in a separate file, so it wasn't that easy to view at the same time. Problem with this was this took a few hours to generate if we were comparing across hundreds of available genomes, and it was also a command line based tool. So the plot itself is something I could share with a plant molecular biologist. They couldn't necessarily run it themselves. And around that time, I was a PhD student still myself. I was annoying the postdocs in the group. I made this is just my recreation of a plot of a slide from back then. But my goal was basically there used to be a photo of an actual postdoc in our group there who was very annoyed with me. I replaced it with an AI representation, but basically I didn't want to be the intermediary. I wanted to make something to be the intermediary, and I could take a rest. But to make this really work is, if this takes several hours still to generate, it doesn't actually make it much easier. I am more rested, but for them, it should really be interactive because they are the subject matter experts and they will want to fiddle with this to basically change the resolution, move around the interval, basically do exploratory data analysis. For exploratory data analysis to be possible or easy, it should be basically very responsive. So this tool had to respond in not hours, hopefully not minutes, maybe even seconds. So I've developed a tool of my own called Tersect Browser, which did just that. So it generates this distance heat map for hundreds to thousands of lung genomes at once. This can be whole genome sequencing or GBS, and then generates associated phylogenetic trees for that. And the users can freely adjust basically zoom level, interval, resolution in real time. And it takes seconds to recalculate instead of hours. There's some extensions as well. If I have time, which I probably won't, I will go into those. But basically, this is all about basically automatically trying to identify introgression signals, jumping to the same position in the genome browser and things like that. So this is the interface of what this looks like. There is an Angular-based interface. Front end, there's a Node backend, but really it's just there to connect to the front end, and then it uses Python scripts and C-based utilities that I wrote myself. And this is deployed and has been deployed for a few years within Cranfield, and we also have been using it with partners. So we made it selectively available to people. Those red bars are gaps in the reference genome. The way this works internally is I've made a C-based utility called Tersect, which is basically a simple C tool for making quick comparisons between contents of VCF files. So basically it creates a very big index using something called a word-aligned hybrid lossless compression. So I can just decompress one part of a chromosome and make a comparison between those. This is word-aligned, so aligned to the processor's word. So I can compare 64 and a 64-bit processor, 64 sites at once. And this is very fast. I'm comparing to very popular tools here. It's very fast, but only for a select few operations. Yeah, it's not fantastic, but it's good for a few operations, including creating distance matrices. And is this fast enough to make my tool respond in seconds? It's not. It would still take about 15, 20 minutes to make that plot. However, I can now pre-calculate, I can partition my genome, pre-calculate bits of it, and then try to recreate the actual interval that the user asked for in the following fashion. So basically, if I ask for, the user asks for from 8 to 37 megabases, it grabs the pre-calculated distance matrix for the 50 megabases, then for 10, subtracts it. This is just Hamming distance. You can just subtract it. And then it finally only makes novel calls using Tersect for the ends of the interval. And this can be quite tuned to run within those few seconds I wanted. And another thing I could take advantage of is when I plot this, if I plot it for the entire chromosome, for example, and the bin size in this heat map is 50 megabases, 50 kilobases, then I would have to draw 900,000 rectangles. So originally I wanted to make like an SVG plot that would be kind of very slow and choppy. So then I thought I could maybe draw this in the canvas, and that's also quite slow. I figured out I can take advantage of the user's GPU just by using CSS, because CSS is already accelerated by the GPU. So basically I draw individual pixels for a heat map and then stretch them out using CSS. If I turn off anti-aliasing, it works. It looks just as fine. Maybe a more mature solution would be to use something like WebGL. Anyway, and now there's all sorts of associated extra features, like if I have an interesting region, I have a synchronized genome browser, and I can just jump straight there and have a look at the genes inside there. I can search for functional notations within that region and things like that. So I'd just like to acknowledge my collaborators, my boss, Fadi Mohareb. I now have a team of MSc students who are working on kind of tidying this up under my direction. And very, very soon, I will be releasing a preprint with this tool to the wider community. Like I said, we've already been using this at Cranfield for a while. I had hoped to make it available to people here and just give you a link, but our IT team told me no. So we'll see about that. We're containerizing it and releasing that. Thank you very much. Please email me. Please email me if you want access just for yourself. That might be easier to arrange.
Thank you very much. I think we have time for one quick question. Is there a question in the audience about the Tersect browser? Okay. I don't see a question, so then I would have a quick question for you.
Yeah.
So you, part of your talk was about scalability, computational scalability, and you also mentioned quickly how you try to achieve that. Have you thought about like visual scalability, like when the elements, the number of elements gets bigger, then it can be also visually challenging to actually see things. Do you use any kind of aggregation schemes, or how do you?
Not at the moment, but I'm sort of revamping it to also take account, for example, for structural variants needs to be blown up and so on. So it needs to be more complicated than it is now.
Okay.
But yes, it's mostly just the CSS for actually not choking on the amount of things that have to be shown. You can always zoom out.
Okay, thank you so much.
Thank you very much.
Again.